Hello, we're glad you've joined us for this live webinar, Easy Automation Solutions, Better Results. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Beckman Coulter Life Sciences. This webinar is actually the first presentation in a three-part series on laboratory automation solutions for smaller labs and those less familiar with automation who are interested in standardizing workflows and saving costs through improved efficiency and results. This year, Beckman Coulter Life Sciences is celebrating 30 years in automation as they continue to provide liquid handling solutions to help accelerate reliable results that drive scientific discovery. Beckman offers a full range of flexible and scalable Biomek-automated workstations and integrated systems to automate genomic, cellular, protein, and other workflows in a wide variety of research areas, including agriculture, cancer and infectious diseases, cardiovascular, forensics, and neurodegenerative disorders. For genomic applications, Biomek-automated workstations are complemented by a growing portfolio of nucleic acid extraction and purification kits built on the company's patented solid-phase reversible immobilization, SPRI, magnetic bead technology. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, David Horvath, MS, and Zach Smith, MS, who are both senior application scientists for Beckman Coulter Life Sciences. David automates proprietary and third-party next-generation sequencing chemistry. He has a Master's of Science in Medical and Molecular Genetics from the Indiana University School of Medicine. David's career experiences span a broad range of life science fields, including forensic DNA analysis, medical diagnostics, and agricultural biotech. Zach automates proprietary and third-party next-generation sequencing kits. He has a Master's in Environmental Science from Indiana University. Previously, Zach worked at the university's Center for Genomics and Bioinformatics as a project scientist. David will now begin his presentation.
Thank you, Judy, and thank you to all of our attendees. I'm glad to have you with us this morning, or I guess evening, depending on where you're at. As Judy mentioned, this is part one of a three-part webinar series, and so today we'll be talking about introducing customers to automating NGS workflows. We'll continue in June with the second part of DNA and RNA sequencing sample prep, and then go into a little more detail on specific methods ranging from simple to complex. The third part of the series is entitled Mission Possible, Automating NGS Sample Prep for Challenging Samples and Niche Applications. So we'll be discussing some of the applications that are becoming available for niche markets and how we address challenges of manual processing for those chemistries. Okay, to begin, we want to talk about what a typical NGS workflow is. And typical is in quotes for a reason, because there really isn't a typical NGS workflow. In general, though, it follows this workflow that we have on the slide. You start with cDNA synthesis for RNA-seq applications, and then continuing from there, you have your traditional library construction steps, which will include end repair, A-tailing, ligation of the adapters, and subsequent cleanups after each of those steps. PCR enrichment is optional for certain workflows, and depending on the workflow, target capture is also an optional part of an NGS workflow. Following the traditional library prep construction, you will continue on to do library QC, quantitation, and sequencing preparation, and the actual sequencing run itself. So although most NGS workflows follow this consistent path, the specific workflow can vary greatly for each application. They can range from single-day short and simple protocols to multi-day highly complex protocols. Even though the workflows can vary greatly, they all share common elements such as reagent and enzyme transfers, sample transfers, feed-based sample cleanup and or size selection, as well as thermocycling and incubation reactions. So we focused on the modularity of those common elements and optimize those and then construct them in different ways to basically assemble the workflows for each individual application. Here we have some workflow examples of a simple NGS workflow for the Rubicon Genomics throughplex plasma-seq chemistry. The protocol takes approximately 3.5 hours to complete. It's performed in a single plate for all the reactions, and there's a limited number of start and stop points. So it's a relatively simple manual process and also a relatively simple application on the automation as well. The first column here, it's a little bit hard to see all the detail, but that's basically your library construction, and the second column is an optional cleanup following your library construction. The next example gets into more of, I guess, a typical workflow as far as complexity is concerned, and this example is for the Illumina TruSeq Nano DNA chemistry. It's approximately a one-day protocol. It involves multiple reactions and cleanup plates and has multiple start and stop points. And to continue on to a difficult or highly complex method example, this is an example of the Illumina TruSeq RNA access method, which is approximately a 3-day protocol. It includes target capture following library construction or adapter ligation, and it has an increasingly larger number of Labware and consumables due to the overall length of the process, and it's highly complex with numerous start and stop points. So again, all these workflows follow a series of reaction setups, incubations, cleanups, etc. It's just the length of the application and the complexity of how those steps are pieced together to create the overall workflow. So if you're already processing if you're already processing NGS samples by hand, you know the challenges of the NGS workflow. So some of those challenges are a significant amount of manual pipetting steps. So with a significant amount of pipetting, you have higher variability, especially with cleanups such as adapter cleanups or your final sample yields. Often it requires skilled pipetting. So what I mean by that is transferring of viscous liquids, in-well tip mixing, or preventing magnetic bead carryover while transferring supernatant off of a plate. Manual pipetting steps can also contribute to user fatigue and injury. Manual processing is highly error-prone. Some examples would be sample and or index transfer mix-ups, sample normalization errors, master mix recipe calculation errors, and pipette volume setting errors. So basically setting your pipette to the wrong volume, affecting reaction chemistry, bead ratios, etc., that are important for cleanup or size selection. In addition, manual processing provides limited throughput and reduced data generation. Small labs, or any lab for that matter, they're always limited in personnel resources, how many people you have to get the work done. Whether you have a small amount of work or a large amount of work, people always are the limiting factor in how much you can get done. Often, there's more effort spent on routine sample processing versus more value-adding data analysis. So automation can minimize or eliminate all of these challenges. So with that being said, Beckman Coulter would like to think that every lab would want to automate, but we hear from customers they're not ready to automate. And here are several reasons that they give as to why they're not ready to make that jump from manual processing to automated processing. We are too small. Well, automation reduces the effort of routine processing and increases productivity even with limited personnel resources. So to allow your small lab with limited number of personnel to be able to do more work more effectively, and again, be able to focus on the value-adding data analysis work versus routine sample processing. Labs also say that their throughput is not high enough to justify automating. Well, throughput is not the only reason for automation. So automation provides more accurate and reproducible sample data, irrespective of sample number, and is scalable to match a lab's growing needs. Some labs feel that they trust their people more than machines. So Biomek-applications are demonstrated and qualified with data to support robust performance. And I'll talk a little bit later about how our development process works for our applications and the types of data that we generate to support those claims and the performance of the application. Budgets. Budgets are always limited, whether you're a small lab or a large lab. So automation can provide significant savings over manual processing through reduced labor or overtime pay, reduced rework caused by human error, and reduced consumable usage via efficient Labware and tip handling. So encourage labs to look at the big picture of how automation can fit into their workflow and provide an overall cost savings as compared to manual processing. And lastly, we don't have the expertise to operate and maintain laboratory automation. So Biomek software is full-featured and easy to use, which makes it the right solution for either the novice or experienced user. Okay, so for the middle portion of the presentation, we're going to talk about what Beckman Coulter does to develop an application. So we're going to start with market research, followed by building collaborative partnerships, scoping the requirements and features of the application, and then begin the development process, where we develop and optimize the method, test the method with real reagents, and generate data, and then produce content such as tech notes, application notes, posters, webinars such as this, which go through an internal and external review process. That's followed by installation of the applications at the customer site, and then post-installation support. So I'll go over some details on each of those parts of our development process. To start with, we have market research. We're looking out into the field to see what applications are in demand, and we gather that feedback from various places, whether that's trade shows, customers, publications, journal articles, et cetera, to see what people are doing in their research. And we utilize that to set a queue up to prioritize which applications we're going to focus on development. When we select the applications to develop, we want to build collaborative partnerships with the chemistry vendors and key research institutions. So they're great partnerships. We work to ensure that the chemistry is being automated in the manner in which it was intended to be run manually. So working with the chemistry vendors to ensure that the data quality comes out automated the same as it does manually, if not better. And we partner with key research institutions, customers who are either using this chemistry already by hand or have an interest in automating this chemistry in their lab. And we use them as a real-world case example for processing these samples in a customer's lab. So we generate data together and ensure that through that partnership that we have a robust and reliable solution to go out to our broader customer base. So after those collaborations are made, the next step is to scope out the requirements and features for the particular application. The goal is to develop a template method that can be readily deployed to customers. A template method is going to be a method that covers the majority of options and workflows that we expect to see out in the field, but still be flexible enough to offer configuration for user-specific needs once it gets out into the field. We focus on things such as throughput, capacity, walk-away time. We consider various accessories and integrated devices that are required to perform the various workflow steps. Workflow options such as sample number, safe stop points, any type of manual interactions for thermocycling that might be needed. All those are considered while scoping out the method, as well as the instrument platform selection. Okay, so I'll talk about our instrument platforms. We have multiple configurations which provide flexible throughput and capability to handle any NGS workflow. The Biomek-4000 is our introductory benchtop model. It's sized just right for low to medium throughput labs. Typically, we develop applications anywhere from 1 to 24 samples on this instrument. While it can handle greater numbers of samples, it's usually limited to that number so that walk-away capability and efficiency is still realized on that platform. It has a pod that has interchangeable pipetting tools, so there's single tools and 8-channel pipetting heads. It also has interchangeable grippers, so you can have the ability to move labware on the deck and not have them be fixed in one spot. The deck layout below is a typical example of our NGS setup for the Biomek-4000. It includes an orbital shaker, static Peltier device for keeping reagents chilled. The next platform is our Biomek NXP, and that's going to be our mid-sized unit. It's capable of medium to high throughput workflows up to 96 samples. It has various pod options. It's a single pod, which can be either a span 8 or a multi-channel pod, the multi-channel having options for either 96 or 384 wellhead. It also has the capability of enhanced multi-selective tip pipetting, which is a multi-channel head that has capability to pipette single rows, single columns, or even individual wells. So it provides some flexibility of a span system while still providing the efficiency of a multi-channel head. There's approximately 16 deck positions in a highly customizable deck, allowing an increase in capacity over the Biomek-4000. And again, an example of our typical NGS deck setup below. With a static Peltier in the back left corner, there's an orbital shaker in the back middle, trash chute on the right, and the two positions on the far left are stacking positions. So that increases capacity for additional tip storage or plate storage. And lastly, we move into our Biomek FXP, which is our largest platform. It's a high throughput, it's a fitting system for high throughput or complex workflows up to 96 samples or more. And I should stress that the size or the amount of throughput you have and the size of your lab doesn't necessarily dictate the size of the instrument that would be right for you. If the workflow requirements for your particular application require a lot of cleanups and a lot of enzyme transfers, then having a dual pod system, regardless of throughput or total capacity, is still beneficial to your lab. So the Biomek FXP can be configured with a combination of 2 pods, a multi-channel 96 or 384 wellhead, as well as a span 8 pod. You can have 2 multi-channels, a multi-channel, and a span, and the option of those. And the deck is highly configurable. It offers a larger deck than the NX or the Biomek-4000 with approximately 20 deck positions. The example NGS deck layout that we have below includes the integrated T-robot, which is our integrated thermocycler on the left. There's a wash station in the back row on the left, which allows for reuse of tips after transfers, an orbital shaker, and then also a static Peltier device to keep reagents chilled as well. Okay, continuing on to scoping options and things that are considered when developing a method, we have options for Labware Selection, heating and cooling, orbital shaking and tip mixing, and tip utilization. So Labware Selection could be tubes versus plates. What's the right labware for your application? It can be based on sample number, dead volume in the tube, what capacity you need for your sample. We have options for reagent reservoirs, whether it's a full plate reservoir. We have modular reservoirs as shown in the center picture below, which can hold different volumes of reagents based on the amount needed for each application. The right modular reservoir is matched to the volume to reduce dead volume. There's necessary magnetic plates for bead separation, with the example picture on the bottom left there. And then thermocycler lids might be another Labware option that's needed with integrated thermocycling on your deck. There's consideration for tip mixing versus shaking. Orbital shaking delivers fast, efficient plate mixing. It reduces tip usage and provides uniform results. Whereas tip mixing works well for larger volumes. It prevents cross-contamination with Labware that's at or near well capacity. So if you have a well that's almost full, orbital shaking isn't the right option for that particular mix step. So tip mixing will be deployed for those purposes. And tip mixing can also access tubes or Labware that are not compatible with orbital deck, with an orbital shaker deck, like a 6x4 aluminum tube rack, a heavy aluminum tube rack that will not fit on our orbital shaker. Heating and cooling options. Peltier units are easy to use. They heat and cool. There's various adapters that can be fit onto the surface of the Peltier to accommodate various Labware, so round bottom wells, flat bottom, PCR adapters, etc. It's an open position, so there's no plate sealing, so considerations have to be made for higher temperatures and evaporation. There's a slower ramping and cooling speed than thermocyclers have, and it's not ideal for all applications, such as high temperatures because of evaporation issues. Thermocycling can be integrated. It can be integrated on all of our platforms, Biomek-4000, NXT, or FXP. And the thermocyclers provide a true walk-away solution, not having to interact with a method for every incubation that you need to perform. And if you're familiar with some of the longer applications, there's multiple incubations that take place, which would require multiple user interventions for off-deck thermocycling. So this allows the instrument to move the plate automatically to the thermocycler and continue the method without any user intervention. The plate sealing lids prevent evaporation, and then the T-robots are compatible with commonly used PCR Labware. And we continue to do development work to integrate additional manufacturers' thermocycling components as well. So tip selection. Choosing the tip type is important depending on the type of reagent you're transferring to, the type of Labware you're trying to access. So our tips come in various volume ranges from 20 microliters up to 1,000 microliters. There's sterile barrier or non-barrier options, and then we also have conductive and non-conductive tips, which are used for liquid level sensing. So tube access is an important part of the selection. The P20 tip is shorter. It has a higher volume without the barrier than 20 microliters. And this might be the right option for a shallow microtiter plate, whereas the P50 is a longer tip. It's more slender, and it would work more effectively with a deep well plate. So after the Labware and the deck layout and all the accessories and active devices are selected for the application, we then start looking at method workflow options. So if you're familiar with our methods, you're familiar with the user interface that we have at the start of the run. If you're not familiar with our methods, all of our methods have a user interface. And so what the user interface does is provide all the option settings that are relevant to the specific application. It's going to have things such as options for on or off-deck thermocycling, options for training mode, which allows you to quickly run through a method without the lengthy incubations that can be used for training, or optimization and testing in your laboratory. It includes options for all relevant application-specific options. That could be, in this case, in the example for the Swift Biosciences method, there's sample input options and fragment size options, as well as which part of the method that you're wishing to run. So in this case, the PCR-free portion of the method has been selected with various index adapter transfer options. All the options are validated for proper selection to ensure that correct inputs are made by the user and will not generate errors upon execution of the run. And there's also safe stopping points built in to these options to allow safe stopping points matching the manufacturer's protocol, ability to safely store your samples at breakpoints, separate safe stopping points for pre and post PCR, being able to stop the method and then transfer your samples to a different instrument if that happens to be the way that your lab's quality management system dictates. It allows for off-deck incubations, and safe stop points allow for recovery by being able to perform portions of the method or go back in the workflow to a previous point and continue from there. And lastly, other considerations that we have in the deck for chemistry-specific issues. There can be multiple issues, but some examples that we have here are considerations for sodium hydroxide. So certain reactions are dependent on the sodium hydroxide to be prepared fresh. So we have built-in considerations for that, where prompts are presented to the user at the right time in the method to present the deck with that particular reagent. For ethanol, we have considerations over evaporation and water adhesion to the ethanol. So we want to ensure that the dead volume is correct for ethanol depending on the length of the run. Walkaway is great, but if you have ethanol on your deck for an entire day, then we definitely have to have considerations for those types of reagents. So after all those considerations are made and scoping is set out, we begin our development and optimization process. So template methods are written to follow the manufacturer's protocol, and then we hit the lab. We have a facility here in Indianapolis with multiple instruments on all of our platforms. We use live chemistry, and the ultimate goal is to generate data with the kits that customers are actually going to use. So to start, we do thorough dry testing and logistical movement testing. We make sure that the method structure is sound, that Labware is moving where it's supposed to be, pipetting is occurring in the well at the right depth, whether that's from the bottom or from the top of the plate. When that all checks out, we move into light reagent testing. And what that is using ethanol or glycerol solutions to mimic enzyme transfers. We're the manufacturer of our bead chemistry that you see in all the NGS kits, so we have the ability to test bead dispensing and accuracy of the bead ratios in-house with our own chemistry. So we use real chemistry when possible during the light reagent testing and optimization. And then we go into optimizing and calibrating pipetting specific to each application or specific transfer. So again, with bead cleanups, size selection, volume transfers and ratios are critical. Same for any type of reaction setup. The balance of the chemistry is critical to the optimal performance of the application. So each of our pipetting techniques can be dialed in to very accurate volume transfers across the range of the pipette tip. So we spend time to make sure that that's set up as accurately as possible to generate the most robust results. Following the optimization, we move into our live chemistry testing. So we've ensured that the method is sound, our pipetting optimization is up to spec, and then we actually run real kits with real chemistry with real samples. If needed, after data is generated, we continue to do optimization that we sometimes don't see until we use the actual chemistry. And we continue to fine-tune the method until we have the results that we're looking for. So we determine those results based on generation of quantitative and qualitative data, such as bioanalyzer tracing, qPCR data, and then downstream into the sequencing data and sequencing metrics. So the next part of our process is content generation and review, and I'm going to hand off to my colleague Zach Smith, and he's going to continue with our development process from this point forward.
Okay. Thank you very much, David. Thank you, everyone, for sticking with us for this long. David's done a pretty good job so far, and I'm hoping to pick up where he left off. So content generation and review process. So that's where we generate the technical and application notes to demonstrate the method capabilities and the robustness of our application. The first thing we'll do is we will have the chemistry vendor go through our data, either our QC traces from the bioanalyzer, qPCR results, or our sequencing metrics, and review those and make sure that the chemistry vendor is content with the results. As David said, we have a facility here in Indianapolis that is fully equipped for this work. We have 2 sequencers in-house, an aluminum MySeq and an XSeq 500, that we can use to generate the sequencing data we need. If we need more data, we have worked on a couple of projects that required high-seq data. We can certainly work with the vendors, or we send those out for additional data analysis. And then we'll work with the bioinformatics groups associated with the reagent vendors to analyze those data and interpret them properly. The data must meet acceptable yields for metrics for yield. The sizing must be accurate, and the sequencing metrics such as the percent rRNA contamination if you're working with an RNA method, or the percent on target for the percent on target reads for a capture method must be consistent with what the reagent vendor is seeing in the manual prep. The other thing we look for is evidence of cross-contamination. So our NGS methods are working with all open plates typically, which helps speed the workflow along because then you're not having to seal and unseal plates. But we do realize it's a possibility that, this represents a risk for cross-contamination. So it's something we're looking for in the sequencing data. And it is something that because we're looking for it in sequencing data, you can see levels that are not detectable on bioanalyzer or other fragment analysis traces. And so we feel like we really do go one step beyond what's typical in proving that our automation solutions are sound and the data, you can trust the data that's being generated. So once that review process is done, then we will generate content for public distribution, like this webinar that you've all kindly decided to show up for. And then we'll move on to application installation. So we realized that while we have we have instrument configurations that we generally suggest for NGS applications, that our customers are working with so much more because our liquid handlers really are application agnostic and they're chemistry agnostic. And so you might be doing NGS applications, you might be doing some cellular or protein work, and so we don't force a customer to buy a specific configuration for their instrument. So we recognize that the applications, therefore, that we develop need to be flexible in their design and that they can be configured properly. So during the course of application installation, our field application specialist will go through and they will implement the method on your instrument, and they will perform a whole range of on-site pipetting optimizations and on-site testing, frequently going through a logistical test to make sure the Labware is moving properly, that your stack offsets are correct, and then they'll go through some pipetting testing and actually go through a complete water run, typically while they're at a customer site, so that you can verify that the pipetting is on target. And then after the application is installed, we have an entire group that provides tech support for these applications, and other groups that provide service support for the hardware. And then we have another third group that provides training and host user group meetings for our customers. So the reason why we do this is it's not like we're just going to go through and airdrop in an application into your site and expect you to just know how to use it. We do backstop our applications with a very extensive group of people that do various varieties of support for our customers. So there's some other additional software features beyond Biomek software that I wanted to touch on during the course of this presentation, both of which are fairly new, Biomek Method Launcher being the newest. But these are software packages that we have developed because we feel like we can offer an improved user experience and better data management and handling during the course of the method being run. So, during the course of the average day, you'll come to your Biomek and you'll say, I want to run a method. All right, well, are we sure we picked the right project? And am I working with the most recent version of my method? Or usually what we find out is our normal Biomek Biomek operator is on vacation and the PI wants me to run an assay and I really need some help with this. So for those reasons and several others, and by the way, this is what our standard user interface looks like. So if you were to open up the editor on your controller, this is what you would see. It's fairly straightforward to use. As far as programming is concerned, the UI gives you a GUI-based interface for building methods and building out software solutions if you want to do that. Alternatively, you can use Biomek-Method Launcher to launch your application that's been installed already. So Biomek-Method Launcher is a new product. It has two components to it. One is the launcher interface, which you're seeing here, and the other one is guided Labware setup. Those are the two primary components here. So the launcher allows you to interact with your instrument without actually running the editor software. So you would just go in and begin Biomek-Method Launcher, and your applications would be listed out there on the launcher, and there would be icons that allow you to select the right one. You can also perform manual control on the instrument through the launcher as well, just by clicking down on the manual control tab, and it will bring up a whole variety of options that you can use to run the instrument. Once you've selected your method, say for example you want to run Illumina TrueSight HLA version 2, an HTML-based user interface would pop up, and it would give you the options you need to run the method, how many samples you want to process, and which components of the method that you want to run. We build these UIs in such a way that they're easy to operate and should be fairly straightforward for any operator to employ. The other component of Biomek-Method Launcher is a new step called guided Labware setup. So guided Labware setup will step you through on a plate-by-plate, tip-box-by-tip-box process to set the deck up in such a way that it is properly set up. And one of the things it really does nicely is that it will go through and will show you side views of stacks of Labware, so you can see that on the right-hand side of your screen. And for reagents that are deployed in troughs, or you have master mixes that you need to create, all those setup instructions are included in the individual Labware descriptions. Additionally, you can print off the guided Labware setup as a PDF, and you can take those setup instructions back to your bench, where you can go and make up your master mixes. The other thing you can do is with those PDFs is those PDFs can be uploaded to a LIMS system, for example, or copied into a lab notebook to show the user, this is what I've done on this particular day. It's a really powerful interface, and I like it quite a lot, actually. So once you get beyond guided Labware setup, then during the course of the method running, this is what the runtime interface would look like for Biomek-Method Launcher. So on the left, you'll see the method screen where it will go through and it will give you a progress bar on the top that will show you how long the method is going to continue. So this answers the vital question of, can I go and get a cup of coffee, or do I have time to go to the bathroom, or is it time to interact with the instrument? And then it will go through and will offer you a series of milestones to let you know what process it's working on and how long that individual process is going to take. If you flip over to the method view over here on the right-hand side of your screen, it will actually go through and show you the Biomek steps that it's actually looking through. And if you go into the Labware dialog, which is on the bottom right of each of those screens, you can actually click and view the volumes on every individual piece of Labware as the method is progressing, so you can monitor its running in real time. Yes, data handling. So automation methods can generate a large number of data that will need to be tracked. Just one well, for example, of this one plate, we could track what the sample ID is, what the project ID is, what the PI is we're working for on this particular project, what species it is, the name of the technician running the assay, concentrations, volumes, the list goes on and on. And the issue gets exponentially worse the more plates you process. It leads to a lot of data that needs to be handled. And of course, it's not just from, this data is piling up during the course of numerous applications. So if you're starting off with NGS samples, either DNA or RNA, you'll want to know, where those samples were accessioned from, any data that's associated with them, including species or the volume or the concentration. Or maybe you'll have to quantify them yourself when you get them in. And so one of the things you can do is you can merge the detector data and track the data and the sample ID to the dilution plate as you quantify and normalize the DNA. Then there's the matter of doing your NGS library prep, and so at that point you'll want to track what samples are associated with what indices so that you can demultiplex samples later. And then you'll want to export some of that data into the Illumina Experiment Manager for your sample sheet. And then also extend that data to the LIMS system if your lab has one for downstream analysis. So DART 2.0 stands for Data Acquisition and Reporting Tool. Is a Biomek is a software package that works with Biomek and SQL to generate a repository that will track all of that information on a per-plate basis. So it can take reader data, it will take data from data sets that are generated as a part of normal operation from Biomek software, and it can report them out to in other devices, a LIMS system, any web-enabled device will allow you to monitor the run in real time, or other terminals inside or outside the lab if you have it configured right. So there's a huge number of ways, there's several ways you can access the data with DART. So you can the DART software create Labware reports that will automatically just expel it into, say, Excel sheets, and then the report contains an embedded link to the database. So you can just click refresh on your Excel sheet, and then it will automatically repopulate that sheet with the fresh data from the report if you ran another assay, for example. So that's really convenient. The other thing you can do is you can monitor runs in real time using the web interface. So DART will report out if the method is running, how long it's been running. You can query all the various sorts of labware and find out what's been done and who's been doing the run. And you can use it to generate all kinds of reports. So on the top left, we've got what the query builder looks like, and you can include any, if you're building a report out, it's basically just a matter of clicking which fields you want to include in the report. And so above, for example, I've got an illustration where you're using DART to report data out to the Illumina Experiment Manager for your sample sheet generation, including the sample and which I5 and I7 indices have been associated with which library. And on the left, on the bottom there, we have a library normalization report from DART 2.0, where we're going through and we're showing what the starting concentration was, how much volume was moved, and what the normalization concentration was afterwards. So it allows you a variety of ways of getting access to your data and storing it. So finally, I wanted to just go over some of our application cards. So we've got a website with a link you can see above, info.beckmancoulter.com/NGS, and there you'll find our current NGS library preparation method card. So this method card will detail what methods we've developed or what applications we've developed on which platforms. This list is continually growing, and we're actively adding content to it it seems like on a daily basis these days. So I strongly encourage any and all of you to go ahead and zip over to this website and for more information. So finally, I just wanted to invite anyone to send us questions via the Q&A tab, and thank you for attending. It's been a pleasure speaking with you.
Thank you, David and Zach, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. David and Zach will answer as many questions as time permits. And the first question is, for the Biomecs capable of the 384 format, is there a 384 well head, or are you pipetting 4 times with a 96 well head? And further, can the 96 head pipette individual volumes?
Okay, so I can answer that question. This is David. The answer is yes. The Biomecs do have options for either a 96 or a 384 well head. As far as the second part of the question, the heads cannot pipette individual volumes as a 96 or 384 well transfer. However, we do have the enhanced selective tip pipetting heads, which are capable of doing individual row, column, or individual well pipetting. So if you did have a 96 or 384 well Labware format that require different volumes in different wells, you could use the enhanced selective tip feature to either deliver or aspirate off different volumes from different wells within that 96 or 384 well Labware.
Thank you. How long does it take to implement an existing method with no modifications versus one with modifications?
Yeah, I can take that. Typically what we found when we install method is that usually there will have to be some sort of modification made to it when it's deployed. We will have a customer want to change which PCR plate that is being used, for example, or they'll have some other requirement. Say, for example, in the case of labs that are working in a clinical environment, they don't want to reuse tips. So we will have to provide a reconfiguration for that. If you had no modifications to make, it's literally a matter of importing the method into your Biomek controller, which takes all of about 30 seconds, and then mapping it onto the customer deck, which again does not take very much time. And then we will go through the process of validating the pipetting and doing the water test run. And that depends largely on the complication or how complex the protocol. So for TruSeq PCR-free with no modifications, it would be approximately 2 days with no changes. But it might be more than that, assuming that we needed to make some modifications.
Can any of the Biomecs process any kind of genomics kits?
Yes, our automation is chemistry agnostic, so we can provide as automation solutions for Illumina, New England Biolabs, Rubicon, Swift, other vendors that may or may not be more boutique. So we also have a variety of extraction chemistries that we sell ourselves, including Formacure, DNA Advance, and RNA Advance, and we can certainly get you more information on those if you're interested.
How do I get support for developing methods not on your list? Do I need to wait until your team provides them?
I can take this one. So we do have a pretty exhaustive list of applications that are demonstrated or qualified. But if you happen to have a requirement for an application that's not on the list, then we do have a field team that covers the U.S. to globally, actually, to be able to handle those specific needs and develop methods out in the field. So a lot of times the voice of customer and the market research that I talked about earlier actually comes from our field team. So they're out working with the customers every day, and method application development could actually start in the field, and it does quite often. So they will develop those methods and then route them through the internal group and allow us to kind of add additional features, the UI options, et cetera, that will then be able to be utilized by many labs. And in addition, the collaborative support that we have for the in-house development of applications extends out to methods that were developed in the field. So if a customer wants a particular vendor's chemistry, field application scientist develops it, then those vendors do partner and work closely with us to ensure that customers' results are optimized for field developed methods as well.
How often do you add methods to your menu?
Seems like weekly. So we're just in the last few weeks, we've added 5 applications to the repository that we source the method card from. So, but our development process is continuous, and it's mostly based on market demand. So we have a focus on providing more solutions for small labs now. So you'll see an increase in the cadence of Biomek 4000 and Biomek NXP methods.
What's the pipetting volume range for your system?
I can handle that one too. So for the Biomek 4000, we can pipette anywhere from 1 to 1,000 microliters, depending on the tip that's loaded and the head that's used. For a 1 microliter transfer for a P20 tip, the precision is the inaccuracy is 5%. For a P1000 tip, for a 1,000 microliter transfer, it's 2% inaccuracy. For our SPAN 8 systems, we can pipette accurately down to half a microliter with an accuracy of approximately 5%. We do actually have a application note around that we can certainly give to people if they're interested in seeing some of the data.
Is there any project to get low retention tips from Beckman?
I can answer that. Basically, the plastic that we use it's always, I guess they call it virgin polystyrene. It's not recycled. And the tips all of our tips are essentially spec'd out to be low retention. And a lot of the pipetting optimization also increases the recovery from samples and liquids within the tip. So for viscous liquids, slower pipetting helps deliver controlled delivery of the viscous liquids, ensures optimal recovery. But again, all of our tips are considered to be low retention.
Can you link the sample tracking to LabGuru?
That's a good question. I'm looking at LabGuru now. Okay. I don't see why not. As long as LabGuru is able to handle CSV formatted files that the sample tracking could generate, I would need to know specifically more about that particular application before I could offer you a solid yes or no.
Okay, give us a moment to see. Let's see, additional questions are flowing in. Hang on 1 second. So what is the sterile barrier of the tips? Is the barrier a filter or other kind of technology?
Yes, I can answer that one. So it is a filter, and it's made of polyethylene, and it does an excellent job of keeping your pipetter, or keeping your liquid handler from getting contaminated by your products. The assembly of the filter is sterilized as a product, so after the barrier is inserted, the whole construct is sterilized.
Very well. So I would like to once again thank David Horvath and Zach Smith for their presentation. Do you have any final comments for us today?
Just thank you everyone for attending. It's been a pleasure speaking with you. And if you have any further questions or comments, please get in touch with us. We'd be happy to hear from you.
All right, and we'd love to have you join us again on parts 2 and part 3 of our webinar series. Again, those are titled DNA and RNA Sequencing Sample Prep for Part 2. That'll take place on Thursday, June 23rd, same time. And Part 3 is entitled Mission Possible, Automating NGS Sample Prep for Challenging Samples and Niche Applications. And that will take place on Thursday, July 28th at the same time, 8 o'clock a.m. Pacific and 11 Eastern.
I'd like to thank the audience for joining us today and for their interesting questions. We'd also like to thank our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. As a reminder, in case some of you missed what the speakers just told you, the webinar is first part of a 3-part series, so save the dates for the upcoming presentation. Part 2, DNA and RNA sequencing sample prep, automating simple to complex methods, will be presented on June 23rd. And Part 3, Mission Possible, automating NGS sample prep for challenging samples and niche applications, will be presented on July 28th. Registration is now open for both of these webinars, so feel free to visit the LabRoots website to confirm your attendance today. This webinar has been approved for continuing education credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through November 19th, 2016. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.